Great. Bismillahirrahmanirrahim. We are going to explain you today the nutritional importance of dietary proteins and protein energy malnutrition. That is, we shall explain you marasmus and kavashir core. In this context, the objectives of our today's lecture are to explain you the, the biochemical functions of protein in the body and to explain you what is amino acid pool and what we mean by the nitrogen balance, and we shall explain you recommended dietary allowances of proteins in the health and in the disease conditions, and dietary allowance of proteins in different age groups. And then we will explain you the role of carbohydrates, that is, what is the protein sparing effect of carbohydrates. And then we shall explain you the protein quality and the biological value of the proteins. And also we shall explain you what are the effects of consuming excess amount of protein in the diet. And then we shall explain you about the protein energy malnutrition, and we shall explain you the differences between the kavashir core and marasmus, and in the end we shall explain you protein malabsorption conditions. In this context, before we explain you the nutritional importance of dietary proteins, first of all, we shall explain you how are proteins different from carbohydrates and lipids, and then we shall explain you the functions of the proteins. As you know, the three macronutrients, that is carbohydrate, protein, and lipids, they are organic compounds. They are consisting of carbon, hydrogen, and oxygen. But in proteins, there is also additional nitrogen. That is 16% of the protein is nitrogen. Therefore, proteins, they are called as nitrogenous organic compounds. Now, as carbons are bonded in carbohydrates, proteins, and lipids, in lipids in different ways, therefore, if we consider the most important functions of carbohydrate, protein, and the lipids, they are different from each other. If we consider the one most important function of carbohydrates, then carbohydrates are considered as best source of energy. Approximately 60% of the energy is obtained from carbohydrates. And if we see the one most important functions of the lipids, then lipids, they are based as stored forms of energy, because we have a specialized connective tissue designed in the body, that is adipose tissue. Subcutaneous tissue is adipose tissue, which is designed to store large amounts of energy in the form of fat in our adipose tissue. Average about 70 kg adult man approximately stores about 11 kilogram of fat in his adipose tissue. Glucose is also stored in the body in the form of glycogen, but there is no specialized tissue in our body for the storage of glycogen. Therefore, limited amount of glycogen are stored in the muscles and in the liver. That is approximately 1 to 2% mass is the glycogen stored in the muscles, and approximately 5% of the liver mass, that is approximately 100 grams of the proteins, is stored in the liver in the form of glycogen. However, proteins are different because there is no storage of protein or amino acid in our body. Though every cell of the body is protein, protoplasm of the cell is protein, but it is not stored proteins. If we consider the one most important function of proteins, then as far as the energy is concerned, The least important function of protein is to give energy. Carbohydrates are the best source of energy. Lipids are best stored form of energy, but the least important function of protein is to give energy. However, the most important function of protein in the body is that it is building block. It takes part in the formation of new tissues, and it takes place and it takes part in the repair of the tissues. So this is the most important function of the proteins. Now, proteins, apart from this most important function, that is, they are building blocks. They take part in the formation of new tissues. They take part in the repair of old tissues. Proteins, as they are composed of amino acids, and there is wide range of functional groups found in the amino acids. These different functional groups include alcoholic group, thiol group, thioester group, carboxylic group, carboxamide group, variety of basic groups like imidazole group, amide group, phenol group. Most of these chemical groups are chemically reactive. These reactive properties are essential for the functioning of the proteins. By these functional groups, proteins can interact with one another and can interact with other macromolecules to form complex assemblies such as histones combined with DNA. These assemblies replicate DNA, transmit signals within cells, and regulate gene expression and carry out many essential functions. Some of the proteins are quite rigid proteins, whereas other display flexibility. Rigid units function as structural elements in the cytoskeleton or in connective tissues, while flexible proteins may act as hinges, springs, or levers and play crucial role in assembling proteins with one another. Like immunoglobulins, if you see the structure of immunoglobulins, they are composed of two light chains and two heavy chains, and they are joined together by means of hinges. Similarly, proteins, they perform many dynamic functions. Proteins uh, play part in the transport, they take part in the metabolic controls, they play a role in the contraction. Proteins take part in the catalysis of chemical transformations. Transport is one of the very important function of the protein, like hemoglobin transports oxygen, myoglobin stores oxygen in the muscles, transferrin transports iron, transcortin transports cortisol, transcobalamin transports vitamin B12. Then we have thyroxine binding globulins, steroid binding globulins, sex hormone binding globulins, vitamin D binding globulins, and retinol binding globulins. So all they play a role in the transportation. Then proteins, they play a role in the blood clotting, like fibrinogen, and rhodopsin plays role in the vision. Then we have the regulatory proteins, which are functioning as repressors and inducers. Then we have many hormones, which are functioning as proteins like insulin, glucagon. There's large variety of hormones. Then we have contractile proteins perform mechanical work. Proteins play important role in movements, both at organ level, like skeletal muscles, heart or gut, and also uh, proteins play a role in movements at the cellular level, like actin, myosin, tubulin plays role in the organization of intracellular contents of the cells, such as microtubules and microfilaments. and they have role in transport, cell motility, and cell division. Similarly, proteins play a role in the digestion of food in the form of enzymes. Proteins, they perform defensive function in the form of immunoglobulins. Proteins, they are precursor of neurotransmitters like dopa, epinephrine, norepinephrine, GABA. Then there are antifreeze proteins present in the blood of Arctic fishes. It lowers freezing point of water, thus saving aqueous plasma from freezing. Similarly, proteins, they perform structural functions. They include collagens, elastins, which form bone matrix, ligaments, and provide structural strength and provide elasticity to organs and vascular system. Alpha keratin form structure of epidermal tissues, nail, hair. So in this form, these proteins, they are performing a wide variety of functions in the body. Now, in this context, apart from so amino acids, we explain you, they are not stored in the body. Hence, they must be obtained from the diet, or they are synthesized de novo, or they are produced from normal protein degradation. Any amino acid in excess of the biosynthetic need of cells are rapidly degraded. In healthy fed state, the total amount of protein in the body remains constant. That is, a person is in nitrogen balance. Now, we will explain you now the turnover of the proteins. So, what we mean by the turnover of the proteins? In this context, it is popular to say that body has no storage deposits of protein, thus adequate amount of protein must be supplied with every meal. However, in actuality, this is not accurate because there is certain percentage of protein in the body that undergoes a constant process of protein breakdown and resynthesis. This process of protein synthesis and degradation is called turnover of the protein. So, turnover of protein, that is synthesis of protein and degradation of body protein, is a normal process, and it is essential feature of what is called nitrogen balance. What is nitrogen balance? Nitrogen balance is important intakes of protein and the amino acid metabolism. So in this context, as turnover of protein, it is the degradation and the synthesis of the proteins. So approximately 100 grams of the amino acids are present in the amino acid pool. Where from these amino acids come in the amino acid pool? What is the source of amino acids, we are saying that approximately 100 grams of the proteins, they are present in the amino acid pool. So these amino acid in the amino acid pool, they are coming from three sources. The first source of the amino acid in the amino acid pool, it is the tissue protein. Approximately 1 to 2% of the tissue proteins are degraded each day, or approximately 3 to 400 grams of the tissue protein, they are degraded each day. These tissue proteins, when they are degraded, they change into amino acids, and these amino acids, they join the amino acid pool of the body. So one most important source of the amino acid in the amino acid pool is the tissue proteins, which are degraded each day. Then the second source of the amino acid in the amino acid pool, it is the dietary proteins. Dietary proteins taken in the diet, when they are digested, they break up into amino acids. These amino acids, when they are absorbed, they join the amino acid pool of the body. And third source of the amino acid pool is the non-essential amino acids. Non-essential amino acids are those which are synthesized in our body. So these non-essential amino acids, when they are synthesized in our body, they join the amino acid pool in the body. So in this way, there are approximately 100 grams of the amino acid in the amino acid pool of the body. And these amino acids from the amino acid pool of the body, then they are utilized. How are they utilized? These uh, proteins, then they are utilized from the amino acid pool, they are utilized for the formation of new tissue proteins. The proteins which are degraded each day, they are replaced with new proteins. Are these amino acids from the amino acid pool, they are utilized for the formation of non-protein nitrogenous compounds. They are used for the formation of porphyrins. They are used for the formation of neurotransmitters. Our amino acids from the amino acid pool are used for the formation of purines, pyrimidines, and other non-protein nitrogenous compounds. And also amino acid from the amino acid pool, if they are in excess, then as you know, some amino acids, they are classified as glucogenic amino acid. That is, amino acid carbon skeleton of some amino acids can be used for the formation of glucose, or can be utilized for the formation of glycogen. That is, carbon skeleton of glucogenic amino acid is converted into pyruvate, or it is converted into other intermediates of the citric acid cycle, which are all glucogenic. Or some of the amino acids, they are ketogenic. They can form acetyl CoA, and acetyl CoA is the precursor of ketone bodies. Acetyl CoA can be converted into fatty acids. So if proteins, they are in excess, then amino acids from the amino excess of the amino acid from the pool is utilized for the formation of glucose, or they are utilized for the formation of ketone bodies, which is alternate source of energy, or they are converted into fatty acids, which are the stored forms of the energy. And these glucose and ketone bodies, they are then oxidized in, in routine way, that is converted into carbon dioxide and water and generate amino acid. So now in this context, the one important point which we have to explain you, we are saying that tissue proteins are degraded. So which of the body proteins undergo degradation? Which of the body protein undergo degradation? Then we have to understand, does all proteins of the body are degraded at same rate or not? If we have to explain you how much proteins are degraded each day and where these degraded proteins go or they are utilized. In this context, we are actually explaining you the turnover of the protein. Now, in this context, how much proteins are degraded? Approximately 100 grams of the amino acid are present in the amino acid pool. If we see the total amount of the proteins in the body, approximately 12 kilograms of protein is present in a 70 kg adult man. From this 12 kilogram, about 3 to 400 grams of the tissue proteins are degraded each day and then they are resynthesized. This is called daily turnover of the proteins. Now, these tissue proteins, they are not degraded at the same rate. Therefore, these tissue proteins, they are divided in two groups. There are two types of tissue proteins. One is the fixed tissue proteins and second is the labile tissue proteins. About half of the body proteins, they are associated with skeleton or other supporting tissues of the body like collagens. Now these proteins associated with skeleton with structures, they have low turnover, and these tissue proteins which are associated with structure or skeleton, they have low turnover in comparison to this other 50% of the tissue proteins, that is called labile pool. These 50% proteins, labile pool proteins, they are mainly intracellular proteins. And these intracellular proteins, they are in more dynamic state, and it is in continuous state of protein synthesis to replace degraded protein. It means these intracellular proteins, they have high turnover rate, or we say they have low half-life. How is half-life defined? What is half-life? The sensitivity of protein degradation is expressed as its half-life. That is, half-life is the time required to lower its concentration to half of the initial value. High rate of protein degradation occurs in those tissues. High rate of degradation occurs. Now we told you intracellular proteins, they have high rate of degradation. Then we say you high rate of protein degradation occurs in those tissues which undergo structural rearrangements. For example, uterine tissues during pregnancy have high rate of degradation, or skeletal muscles during starvation, they have high rate of degradation. Then many proteins which participate in the metabolic regulations, they have also short half-life. A significant amount of newly synthesized proteins are defective due to errors in the translation or due to errors in the misfolding. So these proteins, they are degraded. Even proteins which are normal, when they are synthesized first, they may undergo oxidative damage or may be altered in other ways. In this way, these proteins which are degraded, they must be removed before they accumulate. These proteins which are degraded, they should be removed before they aggregate. Because a number of pathological diseases are such as certain forms of Parkinson's disease, Huntington's disease, they are associated with protein aggregation. So we say most proteins in the body are constantly being synthesized and then degraded, permitting the removal of abnormal or unneeded proteins. So half-life of the proteins vary over a wide range. If we see the half-life of collagens, half-life of collagens is approximately 1000 days. If we see the half-life of lens protein crystalline, it is lifelong, is life of the organism. If we see the half-life of hemoglobin, it is limited to half-life of the red blood cell. If we see the half-life of liver proteins, they range from 30 minutes to 6 days. The half-life of enzyme HMG-CoA reductase, which is regulatory enzyme of cholesterol synthesis, is only 2 hours. Half-life of the heptoglobin is 5 days. Half-life of hemoglobin, heptoglobin complex is only 10 days. Half-life of albumin is 20 to 25 days. So in this way, there is wide variation in the half-life of different proteins. But in general, structural proteins, they have long half-life, and intracellular proteins, they have low half-life. About 75% of the amino acid obtained from hydrolysis of proteins, we told you, they are recaptured. They are used for the formation of these non-protein nitrogens, which are enlisted in the picture. Like porphyrin is found from amino acid glycine. Like you see this creatine, it is formed from glycine, arginine, methionine. Like this purines, they are found from glycine, aspartic acid, and glutamine. Pyrimidines are found from glutamine, aspartic acid. Then tyrosine give rise to formation of neurotransmitters, dopa, norepinephrine, epinephrine, or tryptophan give rise to formation of serotonin. Similarly, there are many other non-protein nitrogenous compounds like glutathione, which is composed of amino acid, glycine, cysteine, and glutamic acid. Carnitine is composed of lysine and methionine. In this way, these amino acids, either 70% they are recaptured for the formation of new tissue proteins, and some are used for the formation of non-protein nitrogenous compounds. And some excess of the amino acids are converted to glucose or glycogen or ketone bodies, or they are converted into fatty acids, which are used. So excess excess amino acids cannot be stored. Surplus amino acids are used as fuel. That is, their carbon skeleton, we told you, is converted into pyruvate, or it is converted into citric acid cycle, and then they take part in the formation of glucose or glycogen. Are they carbon skeleton of excess amino acid is converted to acetyl-CoA, which give rise to formation of ketone bodies. And the nitrogen group of the amino acid is source of urea. So glucose, fatty acids, and ketone bodies can be formed from the amino acids also. So this is the same explanation. That is, carbon skeleton is converted into pyruvate or any other intermediate of the citric acid cycle, where it is source of glucose or source of glycogen, and nitrogen part of the amino acid is converted into. Now, this amino acid pool, where is this amino acid pool present in the body? This amino acid pool does not have This amino acid pool does not have any anatomical reality, or it does not have any anatomical boundaries, but it is present in the intracellular compartment as well as it is present in the extracellular compartment. But this varies with pattern of amino acid in the amino acid pool. It varies with the concentration of the amino acid in the amino acid pool. In general, if we see the concentration of amino acids in the amino acid pool, there is more concentration of amino acid in the intracellular compartment than in the extracellular compartment. This gradient is maintained by active transport. Then if we see the size of gradient, it varies with different amino acids. There is different concentration of amino acid in the amino acid pool. But generally, if we explain, we say 50% of the amino acids present in the amino acid pool is glutamate and the glutamine, which play a role in the transport of ammonia. Then 10% of the amino acid in the pool are the essential amino acid, and remaining 40% are the other amino acids in the amino acid pool. Then coming to nitrogen balance. So what is nitrogen balance? Nitrogen balance is an important index of protein and the amino acid metabolism. Nitrogen balance is used to compare the amount of nitrogen entering in the body and that leaving it. In nitrogen balance, the amount of incoming and outgoing nitrogen is equal. So nitrogen balance is determined by comparing the amount of nitrogen entering in the body and leaving the body. Most of the body proteins are constantly and repeatedly being synthesized and degraded. In adults, this rate of synthesis of protein is just sufficient to replace the proteins that are degraded. So total amount of proteins remain same. This process is called nitrogen balance. So we say a person is in nitrogen balance when nitrogen intake is equal to nitrogen output. So we say a person is in nitrogen balance when nitrogen intake is equal to nitrogen output. And what is positive nitrogen balance? Positive nitrogen balance, it is protein status when dietary nitrogen is retained in the body as tissue proteins. That is, nitrogen intake is more. That is, there is net increase in body proteins. There is net increase in the body protein than nitrogen output. When nitrogen intake is greater than nitrogen output. And this is seen during positive nitrogen balance is seen during the condition of growth, during pregnancy, convalescence, tissue repair after injury, recovery from illness, and increased physical activity. Hence, all these conditions required more amount of the dietary protein. So in general, positive nitrogen balance is that when there is more retention of nitrogen than nitrogen excretion. Then coming to negative nitrogen balance. What is negative nitrogen balance is when output of nitrogen exceeds the nitrogen intake. Nitrogen intake is less than the nitrogen output when there is more loss of nitrogen than nitrogen intake. It results from an inadequate dietary intake of protein. And this negative nitrogen balance, it can be seen following the surgery in conditions of advanced cancer, in conditions of debilitating illness, wasting diseases in old ages, and following failure to ingest adequate are sufficiently high quality proteins, as in proteins, or it is seen in the protein malnutrition like kwashiorkor and in the marasmus. Then coming to protein requirement in the body. The recommended dietary allowance of protein is approximately 0.8 grams per kg body weight per day. As you know, proteins are available to us from plant sources like legumes, pulses, and from the animal meat. So it is also known to us that proteins which are obtained from animal sources are good quality as compared to plant sources. In this context, we are saying that recommended dietary allowances of proteins depend upon many factors. That is, depend upon the protein source, depend upon the protein quality, depend upon the age, depend upon the sex, and depends upon the physiological status, whether it is condition of pregnancy or lactation, and depends upon the health status, either present, either a person is suffering from any trauma or infection or burns or carcinoma, or and we have to see whether this person is taking sufficient quantity of carbohydrate or fat in the diet, because uh, carbohydrates also have protein sparing effects. So in this context, we will say what are right and what is dietary allowance recommended, how we define recommended dietary allowances. Recommended dietary allowance is the amount of nutrition required to meet the physiological need of an individual. Our recommended dietary allowance refers the amount of nutrient that is required for optimizing the health. Recommended dietary allowance is not the amount of nutrient required merely to avoid the disease. So in this context, we are saying recommended dietary allowances of protein of mixed biological value is about 0.8 grams per kg body weight. That is, 70 kg man requires approximately 56 grams of proteins per day. But this requirement, 56 grams, may be increased. These recommended need to be increased in the person on the vegetarian diet, because plant products are much less in proteins than the animal proteins, and vegetarian diet has much less caloric densities than the animal proteins like meat. So in general, animal proteins are said of high biological value. However, gelatin, gelatin is prepared from the animal collagens. It is exception. Collagen, derived protein from the animal protein, it lacks all the essential amino acids. But in generally, animal proteins, they have they are good quality proteins. Proteins obtained from the plant sources have lower biological value because they lack in one or more essential amino acids. Missing amino acids uh, in the plant sources are called the limiting amino acids. So, to overcome this limitation, foods are consumed in combination. For example, wheat protein is deficient in lysine and contains other essential amino acids in sufficient amount. But kidney beans, they contain lysine in sufficient amount. So when wheat proteins are mixed with kidney proteins, uh, then this deficiency of essential amino acid is recovered. Now, as you know, the proteins, they are required for the formation of new tissues, and they are required for the maintenance or repair of the tissues. Therefore, requirement of protein is increased in the diet in the periods of rapid growth, such as there is increased requirement of proteins in condition of pregnancy or in infancy, childhood, or adolescence. So a young child may require 2 to 3 times the of that amount. So children should consume 2 grams per kg body weight per day. A pregnant woman needs additional 10 grams of protein, and lactating woman needs extra 15 grams of protein per day. Then coming to illness, protein requirement is increased in highly catabolic hospitalized patients because there is loss of body proteins in response to trauma and infections. One of the normal metabolic response to infection or trauma, such as burns, a broken limb, or surgery, is increase in the net catabolism. There is increased net catabolism in the traumatic conditions due to action of cytokines or due to action of hormones like norepinephrine, epinephrine, glucagon, cortisol, which when are released, they cause lipolysis, proteolysis, and cause gluconeogenesis. As a result, there is excessive utilization of amino acid. Therefore, there is increased requirement of proteins in the condition of illness. Again, coming to protein sparing effect of the carbohydrates. So we say another factor that must be considered in determining the protein requirement is the dietary intake of carbohydrate and fat. If carbohydrate and fat are present in insufficient quantities, then what happens? Some dietary proteins must be used for energy generation. So apart from their primary function of formation of tissues and repair of tissues, these amino acids, if carbohydrate and fat is not available, they are used for energy generation. You see, the daily minimum requirement of carbohydrate in diet is about 150 grams per day to avoid ketosis. Because so approximately 150 grams of carbohydrate is required per day. 100 grams or 150 grams of fat are required to provide us essential amino acids and fat soluble vitamins. So you see, an adult brain utilizes about 120 grams of glucose is utilized only by the brain per day. And in addition to these, those tissues which have few number of mitochondria, they also undergo anaerobic glycolysis, like red blood cell, which lack mitochondria. The cornea, lens, regions of retina has limited blood supply, and they lack mitochondria. Medulla of kidney, testes, leukocytes, white muscle fibers, all depend upon glucose because they have few number of mitochondria. So when carbohydrate intake is less than 130 grams per day, amino acids are deaminated. Then amino acids are deaminated to provide carbon skeleton. Amino group is converted into urea, and carbon skeleton of the amino acids. In this condition, when carbohydrate is not available, when it is less than 130 gram, this carbon skeleton is used for glucose and gluconeogenesis. Thus, protein requirement is increased. So, as energy content of diet from carbohydrate and fat increase, if energy content of the diet from carbohydrate and fat increase, then the need of protein is decreased. So dietary carbohydrates is somewhat more efficient protein sparing effect. So now we come to why we need dietary proteins. We need dietary proteins for body growth, repair of tissues, and for the maintenance of the tissues. Dietary proteins, they replenish the amino acid lost in the urine, feces, saliva, skin, hair, and nail. And approximately 1 to 2 gram of nitrogen is lost as undigested protein, which is excreted in the feces. We need dietary proteins to replenish amino acid in the amino acid pool. After these amino acids are being used for synthesis of non-protein nitrogenous compounds and for other body activities. And we need dietary proteins for maintenance of output of essential secretions, digestive enzymes, and synthesis of the plasma proteins. So in this way, these are the, for all these purposes, we need the dietary proteins. Then coming to protein quality. Proteins obtained from different sources, they differ in their ability to support growth, to maintain repair of body tissue. This ability is indicator of quality of the protein. So proteins obtained from different sources, they differ in their ability to support growth, to maintain the repair of tissues. This ability of protein is called, this ability is indicator of protein quality. So it means, you see, it depends upon the amino acid composition. So protein quality depends upon the amino acid composition, whether it is containing the all the essential amino acids or not. This is 9 are the essential amino acids: leucine, isoleucine, lysine, phenylalanine, tryptophan, threonine, methionine, valine, histidine. These are the essential amino acids. And these essential amino acids, are defined as those amino acids which are not synthesized in the body and they are taken in the diet. So quality of the proteins depend upon the amino acid composition, whether they are rich in the essential amino acids or not. Then it depends upon the digestibility of the protein. That is the protein which is taken in the diet, either it is digested or not. Like you see, bone proteins, collagens, they are not digested. So if protein is not digested, though it contains essential amino acids, they are not available to us. Then quality of the protein depends upon the efficiency with which amino acids they are absorbed and they are subsequently incorporated for the formation of body tissues and for the repair of the tissues. So in this context, what is biological value of protein? So one of the important parameters used to define the protein quality is biological value of protein. What is biological value of protein? Biological value of protein is the percentage of absorbed nitrogen retained in the body. Retained in the body. So biological value of protein, it is the amount of nitrogen retained and the amount of nitrogen absorbed multiplied by 100. This is actually ability of a protein to provide essential amino acid to the body, which is in proportion to body needs. Now, for example, biological value of egg protein is 96 because it contains sufficient amount of all the essential amino acids. Generally, animal proteins, they have high biological value than the plant proteins. Then coming to excretion of nitrogen. As we told you, proteins are nitrogenous organic compounds and 100 gram and 16% of the protein is nitrogen. It means 100 grams of the dietary proteins contains approximately 16 grams of nitrogen. Now this protein, when it is utilized, its nitrogen is excreted in different forms. It is excreted in the form of urea. Nitrogen is excreted in the form of ammonium ions, or it is excreted in the form of uric acid. So 83% of the nitrogen is excreted in the form of urea. 70% of the nitrogen is lost as ammonium ions, and 10% is lost as organic waste that is lost in the form of uric acid and creatinine. Most of the nitrogen is eliminated from the body in the urine, and approximately 1 to 2 grams of nitrogen is lost as undigested protein and excreted in the feces. Then what happens when excess amount of the proteins is consumed than the recommended dietary allowances? Recommended dietary allowances, we told you it is 0.8 gram per kg per body weight. Our 70 kg man requires 56 grams of the protein per day. So in this context, much is said about the consumption of large amount of protein. An average American is consuming approximately 99 grams of protein, out of which 68% is from the animal sources. So actually, the proteins which are consumed in excess of the body needs, then they undergo deamination.